Okay, hello everyone. Welcome back to the sixth episode of the Harvard Medical AI speaker series. And today we have Thomas Buckley, who's a PhD student at the AI and Medicine track in Biomedical Informatics at Harvard Medical School, advised by Professor Rajman Rai. His work focuses on developing and evaluating LLMs for clinical reasoning and diagnosis. And today he's going to talk about his very recent preprint titled Navigating Gigapixel Pathology Images with Large Multimodal Models. So thank you, Thomas, for joining us, and take it away.
Thank you, Mohammed. Today I'm going to be presenting work I did with my colleague Keon, who's a PhD student at MIT, um, where we try to augment large language models with very large gigapixel pathology images. So the challenge here is we want to be able to ask general questions about a whole slide image in pathology. So when you're working in pathology, you have tissue samples, which are placed on a slide, and you view them under the microscope, and they're extremely large. So each dimension can have up to 100,000 pixels. Um, there's really kind of detailed information that you would want to see. Um, for example, you would want to look at cells at a really high magnification, but you also need to have knowledge of the entire slide at kind of a lower magnification. So there's this spatial hierarchy that these images have that makes it hard to just say put this image into a normal um, machine learning model. Um, so we'd like to be able to leverage models like GPT-5, which are very strong reasoners. They're generally good at answering kind of visual question answering tasks. It would be nice if we could use these in pathology, but previously they've shown very poor performance on this task, in part due to the size of these images and a lack of training data. Um, So the current best approaches are you either train kind of a domain specific model using a lot of pathology data. So at the top here, I'm showing how this model PathChat was trained. So this was published in Nature 2 years ago by Faisal Mahmood's lab, and they trained a lava style model on millions of image caption pairs in pathology. And this model ended up doing really well, but a primary limitation with it is you can't provide the model a whole slide image directly. You have to pre-select a region of the slide, which requires some expertise in the first place to do this. Um, Alternatively, if you want to use a model like PathChat or like GPT-4 with a whole slide image, what's typically done is you either give the model a low resolution thumbnail of the image, or you randomly select patches of the image. So you'll segment the tissue and then sample like 50 patches, for example, and give each one of them to a separate LLM call, and then aggregate the responses either by majority vote or using another LLM. Um, so that's essentially the best baselines that these models are compared to in the literature. Um, Another approach that's more recently been done is training a lava style model, but using embeddings of the whole slide image itself. So this was a paper in CVPR last year called Slide Chat, um, where they took 3,000 images from TCGA. They generated embeddings of the whole slide images using a patch level encoder and then a slide level encoder. And then they trained this lava style model using captions that were generated by another LLM. Um, and this they showed that this works pretty well on images within TCGA within their test set. Um, but we were wondering like how general is this approach? Can you ask what kind of questions can you ask a model like this about a pathology image? Um, And then, of course, this paper compared to, like I said, they compared to GPT-4, provided random patches or provided a low resolution thumbnail, and they showed that their model did a lot better, similar to other papers. And we saw this, and we're essentially wondering, like, how can this possibly be the best baseline of a model like GPT-4? Because these images have intricate spatial hierarchies, right? If you just give it a low resolution thumbnail, you're missing a ton of information, a ton of really important diagnostic information. Shouldn't really be possible to give a diagnosis based on just a low resolution thumbnail of one of these images. Um, The other surprising thing is you see this slide chat model. It's better than these other models, but not by a lot. Like, it's only 10% better on one of these tasks, and at most it's 15% better, which is you'd expect something a lot higher given how bad these baselines are. Um, so we were wondering, is this a good benchmark as well? Like, are these questions actually challenging? Do they require the level of analysis that a human pathologist would typically offer, where they're going to scan the slide at multiple magnifications and do a really detailed analysis of the slide? Um, So some other models that we tried to evaluate were also Titan, which is a large model also from Faisal's lab that's trained on millions of whole site images, and it's a clip style model, and you can do zero shot classification with whole site images. And then these others here I kind of discussed. There's also SlideSeek, in which another paper from Faisal Mahmood's lab. They're doing most of the work in this area. They took an OpenAI O3 large language model and gave it PathChat as a tool. So the model can look at a whole site image thumbnail, select a few regions it's interested in, and then get a response from PathChat. And the author showed that worked pretty well. But essentially, my argument here is that they didn't do kind of the baseline comparison of how does a model like OpenAI O3 do on its own without being augmented with a tool. That's still kind of unknown. Like, is there a way that we can build a scaffold around an existing LLM to enhance its performance beyond just doing the naive thing of giving it a low resolution thumbnail or giving it random patches? So what we implemented in this paper we call Giant, which stands for Gigapixel Image Agent for Navigating Tissue. And what we do is we build this kind of simple scaffold where we give a large multimodal model, for example, GPT-5, we give it the thumbnail of a whole site image, but then we also equip it with this tool called Crop Tool, which lets the model select a region it wants to see on the slide. And then we iteratively give it that region. So this runs in a loop from anywhere from 5 to 25 iterations, and the model can just repeatedly select regions of the slide it wants to scan, reason about that region, and continue kind of looking around the slide. And we hypothesized that this would do a little better than the thumbnail or the patch approach because the model can choose which kind of regions of the slide would it be important to analyze and explore the slide much better. So an example of what this looks like, if we ask it a question like, what is the diagnosis? We give it the thumbnail. The model generates some reasoning. So this is a reasoning model. We append this question, this first image, the reasoning, and then the tool call into the context window of the LLM. And then we ask it again to make another tool call if it wants. So I have a demo here. We built this like kind of simple UI for the model. So you can ask it any question. You can ask it anything you would ask GPT-5. I'm asking here, what is the diagnosis? We give it the thumbnail, which is very large. We also add these like crosshairs to the image to give the model a sense of the spatial coordinates, like where it should make tool calls. So we can see it's kind of iteratively selecting regions. This is clearly a higher magnification than the first thumbnail. We don't see all the reasoning from the model because OpenAI hides some of the reasoning. And then you can see here the model starts generating some reasoning on step 3. I'm looking to identify the orientation of fat or epidermis at the edges. I want to zoom out for a broader view. So we can see the model's acting like fairly intelligently here. It's both zooming in and zooming out and collecting a lot of information about this slide, which we wouldn't have had previously. So great, we have this kind of model, and the question is, how do we evaluate something like this? There's not really a great existing benchmark for asking general questions about a whole site image. So we broke it down into kind of 4 basic tasks that would be expected of like a professional pathologist or even a med student studying pathology. So the first one is general question answering. So given this slide, can you ask a question, for example, like what what quadrant, what region of this image is the metastasis in, what's the depth of invasion of the tumor in this image, for example, like questions like that. Even simple questions like how many pieces of tissue are in this slide. Some other ones are tissue classification. So given the slide, can you identify what organ it is? Is it heart? Is it lung? These are based on normal tissue. We wanted to see if it can do cancer diagnosis and then also cancer grading. So given that the slide has cancer, what grade is it? So to create a benchmark like this, we collected slides from a few different publicly available repositories. The first one was TCGA, which is a set of cancer images. The second one was GTEx, which is contains actually a large set of normal tissue images. And we wanted to use this to create a benchmark of can the model identify the type of tissue on the slide. And then finally, we pulled images from the Panda Challenge, which is a benchmark of given a prostate tissue image, can the model provide the right ISUP score, which is a cancer grading score from kind of less aggressive to more aggressive cancer on the slide, or essentially the stage of the cancer on the slide. And then finally, we created a new benchmark because there was really only one existing benchmark for question answering over whole slide images, and it was generated using GPT-5 provided reports from TCGA. And we were kind of concerned about the validity of this benchmark. So we created a new one where we took 90 whole slide images from TCGA, and then we asked 2 professional pathologists, 1 attending, 1 resident, to generate questions, either easy, medium, or difficult questions about the whole slide image that kind of reflect what they do in their day-to-day practice. And we use this to generate a new benchmark of much more challenging questions using these images. So some example tasks are organ classification, cancer diagnosis, cancer grading. This is an example from SlideBench VQA, which is the benchmark I mentioned that was generated for the SlideBench paper. One of the questions is like, what type of inflammatory change accompanies the tumor in this case? And there's multiple choice options. And then we created this expert VQA benchmark. For example, you may ask, identify the tumor's maximum depth of invasion within the tissue, and you get multiple choice options. So the first thing we tested was the expert VQA benchmark and the SlideBench VQA benchmark of our model, so GPT-5 equipped with this giant scaffold, versus Titan, which is a zero-shot model, and SlideChat. We found that using the scaffold is a lot better than the prior baselines in the literature, especially on this new benchmark of realistic clinical questions we created. And it outperforms both Titan and SlideChat, which is pretty interesting given SlideChat is trained on TCGA. These are also TCGA images. The questions are just maybe more out of distribution for SlideChat than what it was trained on. On the benchmark from the SlideChat paper, this was kind of expected, but SlideChat does better on these questions. But we're still able to outperform Titan, which is kind of the largest zero-shot model available for this task. When we look at the much more challenging classification tasks for across organ classification, cancer diagnosis, and cancer grading, you can do a lot better with just GPT-5 and this giant scaffold than using SlideChat, which is the best existing whole slide image chat model. At the same time, you do quite a lot worse than using something like Titan, which is trained for this purpose. So you can essentially do better on some tasks and worse on others. We tried using other base models. We tried using GPT-4o and Claude 4.5 Sonnet, and they do a lot worse than GPT-5. GPT-4, when you give it the patch and the thumbnail, basically refuse to answer a lot of the questions in the first place. Like it won't. If you give it a thumbnail of an image from TCG and ask for the cancer diagnosis, it's not going to give you the answer, because the model has some understanding that this is not even a possible task you can give it. But we're seeing kind of decent performance with GPT-5. So this is the first model that seems to be able to do this task pretty well. We also looked at how does this change with the number of iterations we allow the model. Um, Interestingly, we found that performance kind of saturated after 5 iterations. It gets better for some more challenging tasks. So for TCGA cancer diagnosis, it looks like scaling continues up to 20 iterations, which makes sense given this is much more challenging and maybe the model needs to see more of the slide. Um, but for the other tasks, which are a bit simpler, the performance saturates fairly quickly. We also did an ablation of does the model making these deliberate choices of where to scan on the slide, does that actually matter at all? Or can we just give it a bunch of random regions in the slide that have tissue on them? So we compared our agent selecting a patch each time to just giving the model random patches at random magnifications that have tissue on them. Um, and we found that letting the model choose does a bit better than giving it these random slides. So it does appear that it's actually important to give the model a tool and let it choose which region it wants to see next. We also tried equipping the model with a specialist tool. So there's been a lot of research on this. So I showed SlideSeek, which equipped GPT-5 or it equipped OpenAI o3 with a PathChat tool. We don't have access to PathChat, so we used like the second best thing, which is Conch, which is another clip style model, but it uses ROI. So you can give it a region of a pathology image. You can give it a bunch of class labels and get essentially the most likely class for that image. Uh, we found that the model surprisingly doesn't do much better. The results were very mixed. Um, we found essentially that the model would very quickly narrow in on a set of either diagnoses or answers that it thought were correct, and it would use the tool to kind of confirm its biases or not really listen to the tool when it gave it contradictory information. Um, so I think this is an interesting result, and I think it highlights that this problem is not so easy as just giving GPT-5 a specialist tool. There's a more fundamental issue of the model having a bias here, and not being able to correctly distinguish between visual features on the image. Um, Finally, we wanted to kind of quantify the failure modes of this system. So we took a small subset of 28 images that were randomly either cancer or benign, and then we asked this giant system, what is the organ type and what is the diagnosis? And we didn't give it any multiple choice options. We wanted to just see how it would reason about the image. And then we gave these to an attending pathologist and asked it to look at each step of the reasoning and see if they agree with, the model's explanations for why did it zoom to the region, what did it see in the region, and how does that relate to the final diagnosis. And we found that the model is best at understanding why it would zoom to a region, but it's much worse at actually uh, understanding the visual features it's looking at and how they relate to a final diagnosis. So, for example, it frequently uh, call a bunch of different cell types skin. It would just think everything it's looking at is skin. Um, so there's clearly some issues with GPT-5 as a model looking at pathology images. So there's just a lack of maybe pathology training for a model like GPT-5 that's going to limit the performance you can get out of a system like this. Um, so yeah, that's essentially the whole paper. If you want to read the full preprint, I have the archive link here. And then you can, email me if you have any other questions, and I'm happy to answer questions now.
Thank you so much, Thomas, for the talk. We have some time for questions. I have a couple of questions I can start with. So first thing, the crop tool that you guys used, is it the same the one that GPT already has access to? Because I think it already has access to a crop tool, right? Or is it a different one that you guys gave it access to?
Yeah, so when you give it an image on chatgpt.com, I think this is what you're referring to. It will
Yeah.
like in its reasoning crop the image. Um, we were definitely inspired by that. I'm not exactly sure how I think they implement that just using like a Python tool. Um, so ours is implemented a bit differently because we had to take into consideration that, um, you can't really like directly crop a whole slide image because it's too big. Um, you have to take the region and essentially find what is the highest resolution magnification that would result in this crop. We had to do like a little modification, but it's essentially the same tool.
And it can do different cropping of different sizes, or is it just uh,
Yeah, so you can specify any XY coordinate, any width, and height, and it'll give you the image. We just constrain it such that the maximum, after it does that crop, the image will only be at most 2000 pixels on either edge. So it finds the best magnification that will give you that image. Um, these images are typically stored in multiple magnifications just because they're so big. Um, so you just step down the magnification until you get an image that's big enough for that.
yeah, that makes sense. And then on slide 13, how different is the expert VQA dataset versus the slide bench? Like, what are some uh, questions that are in expert VQA but not in slide bench? And is slide bench also made by are the questions, the VQA questions, also made by pathologists, or is it? Uh,
Yeah, that's a good question. So SlideBench was the one created using TCGA reports and then having an LLM generate multiple choice questions from it. So there was no pathologist involvement in SlideBench VQA. Um, the questions in our data set are actually fairly similar. We actually gave the pathologists examples from SlideBench VQA to help them create questions. I think the primary reason performance of SlideChat is so inflated on SlideBench VQA is that um, they train the model on questions that were generated using the same process as questions in the test set. So I think their model got very good at answering questions kind of similar to whatever the model was generating. Like there's very little variance within this benchmark of the types of questions that are being asked. Um, I think our benchmark, it's hard to quantify how different it is, but we do make it publicly available. So if you want to look at kind of the qualitative differences, you can see that.
I would assume if you provide the model the report from uh, the TCGA dataset and make it answer questions from expert VQA, would it be able to answer it, or?
Yeah, that's a good question. Probably not because we didn't give the pathologist the report or even the label. We just had them look at the image and kind of interpret the image on their own using their expertise and generate a question about the image. So there wasn't any constraint that these questions would be found in the report. So it's actually very likely that if the model had the report, it wouldn't really help it with these questions.
My last question there is in regards to augmenting GPT-5 with a tool, like a specialized pathology tool. There's a paper called Towards Generalizable AI in Medicine via Generalist Specialist Collaboration. And what they do is basically they use GPT as an orchestrator, and then they don't just use one specialized model. They have like 10 different ones. So you can have like Coach, what's it called, Conchi by
Yeah, conch.
Conch, um, and then a bunch of others, and then you can make the orchestrator GPT take in all of the predictions from each model, and then tell it to uh, use that to help its final prediction. So something like that could also be done here.
Yeah, that's what we're trying to do now. Um, yeah, I think it's just more challenging than we expected. I think because the model kind of alluded to this earlier, but it kind of comes in with some biases around the initial image that you give it, and it's sometimes difficult for the model to like change its mind, for example, when it sees new information from the tool. Um, so we're trying to find a way to make this work. But then another thing is a lot of these specialist pathology models aren't publicly available. So PathChat, Titan, you can't actually use to generate reports. They don't release that. So there's the amount of specialist models that are actually decent, or it's essentially just conch um, at the moment. So I think until we can get more of these specialist models, it's quite hard to do this.
That's weird. I would have thought there is a bunch because every now and then I see like an open source uh, pathology foundation model being released.
Yeah, yeah. So like Titan is open source, but I think because the reports are they include like patient information, they were only able to open source the like embedding model, so you can't use it for report generation.
Okay, sorry, one last
Um,
question before uh, others can jump in. Slide 15, I think it was, where you're showing the tables of, yeah, this one. Why is why is slide chat so bad on this, uh, on the first task specifically?
yeah, so when you ask slide chat to answer this like 30 class classification task, um, we tried a lot of different prompts. We tried a lot of different ways to make it work. The model will essentially just give the same answer for every single image. Um, so I think slide chat was not trained very well. It doesn't really generalize to anything other than multiple choice questions that look really similar to the ones it was trained on. If you ask it anything unlike that, if you give it even like five options instead of four options, it kind of breaks down.
I was probably just then fine-tuned on just one type of prompt or something to where.
Yeah, no, they only fine-tuned it on these four option multiple choice questions. And we didn't really want to do any further fine-tuning in this paper because the point was like, can you use a generalist model without any fine-tuning? Um, so there probably is opportunity there to take something like a slide chat architecture and maybe train on more data, use a bigger model. I think you could definitely do better than this.
If there's any other questions, people can jump in.
I had a question really quick.
Yeah, yeah, sure.
Um, Great talk, first of all. I thought it was really interesting and I thought it was a really unique approach. Um, could you go back to the slide that like shows all of the different tasks, the different benchmarks?
Was it this one?
Yeah, this one here. So I was curious, like this isn't like your paper, but do you think that there's any like sort of reasonable concern that some of these data sets, like for example, like TCGA or like Panda, and their associated reports or labels, like could be included in like GPT-5 training data? And if they're like, if that sort of reduces like the validity of them as like benchmarks in this task?
Yeah, I think it's a fair concern. It kind of went into our design of the expert VQA benchmark. As yeah, it may be true that the reports are in the data set, but it's pretty unlikely that novel questions generated by pathologists without the report would also be memorized in some way.
Right.
Um, I think the other thing is like the reports are pretty sparse. I'm not sure they're in the training data. We could definitely check for that. Um, but it's pretty unlikely that there's any chance that OpenAI is training on these like whole slide images directly. I'm not sure how they would do that. But it's definitely a real concern. I think it would be good future work to kind of probe the model and see if it has memorized any of these reports. But I think at the moment we think it's pretty unlikely.
Okay, gotcha. Yeah, because even though it's like it's unrealistic for them to maybe train on like the whole slide image, even like if the model like recognizes the thumbnail from like a particular data set or something, I was thinking maybe it might be able to tie it back to that. But
Yeah, I
yeah,
think
I
it's a
agree.
fair concern. I think it's definitely fair that if you ask the model for like a diagnosis and you give it the thumbnail and it can tie it back to the report and give you the diagnosis, like yeah, I think that's definitely possible. The questions in expert VQA are more about like the morphology of the slide, which I think are unlikely to appear in like a report.
Right. Okay, gotcha. Thank you.
Great. Well, thank you. I guess there's not any more questions.
Um, If there's no other questions, we can stop here. Thank you so much, Thomas, for joining us today. It was an amazing talk. I thought the approach is quite unique, and I enjoyed learning about it. And pathology is not my field, so there's a lot of stuff that I learned today that I didn't know about. But yeah,
Yeah,
thank you so
thank
much
you
for
for the
coming.
invite.
Of course, of course. Keep up the good work at Malai's lab, and see you later.
Yeah, thank you. Bye.
Bye, everyone.